Good morning, Miss White here. Welcome to part one of the lecture on meiosis and sources of genetic variation. And as you watch this video, make sure that you have your lecture packet out, something to write with. Remember that you should pause the video at any time to write down notes on your lecture packet, or switch over to Canvas to watch any animations that are related. And please make sure you write down questions that you want to ask. You can either ask them in the comments or please save them to ask in class. All right, let's get started. Okay, these are the learning objectives that we're going to address in this lecture. We'll take it part by part, but you can see we're going to go over learning objectives 1 through 5. These are also written on your lesson calendar, and these are the ones you need to study for the quiz that is due before we come back from break. So the first thing I'd like you guys to do is pause the video, brainstorm a little bit. We want to know some differences between a life cycle that involves asexual reproduction versus one that involves sexual reproduction. So think a little bit, write some things down, and then join me in a minute. Okay, hopefully you have some things down, so let's go over them. One of the things that we want to make sure you know is that when an organism is going through an asexually reproductive cycle, the offspring will be an exact copy of the parent. We can call them clones. However, if it's a sexually reproducing life cycle, the offspring will be genetically different. So we're going to be looking at how that's possible. In the asexual reproduction, you only need one parent because one parent becomes the daughter cells. It divides in two, as we saw with mitosis. In sexually reproducing organisms, it's two parents that are needed in order to contribute the two sets of genetic information. You might see these abbreviations because in an asexually reproducing organism, it can actually start out as haploid. So make sure you know that when you see one N or just N, it represents a haploid cell. If it's making a clone of itself, it will remain haploid. If it starts out diploid, it's going to remain diploid. On the other hand, in the sexually reproducing organisms, there will be an alternating haploid and diploid stage to the life cycle. The organism may start out diploid. It's going to become haploid through the process of meiosis, and those would be the gametes. And then when those two gametes, a sperm and an egg, combine, it will become diploid again. So you get this alternating haploid and diploid stage. Now we're going to see asexually reproducing organisms undergo mitosis. That would be in eukaryotes. And if it is binary fission, that's going to be prokaryotes or bacteria, the simpler process. Okay, those are prokaryotes. All right, whereas in order to produce gametes necessary for sexual reproduction, the cells need to undergo a new process of division called meiosis that we're going to be learning about. Okay, so as I mentioned, all sexually reproducing organisms have alternating diploid and haploid stages, and we are most familiar with the animal life cycle. Here's an example where the haploid stage is a very small portion of the life cycle. The majority of the mature organism is diploid. See the 2N right there? That would be all of our body cells. And then there's a small portion of our cells that are going to undergo meiosis to produce the gametes. So we still do have an alternating life cycle of diploid, haploid, but the majority of our cells are diploid. Let's look at this diagram together, and then I want you to pause the video and write down a description of what you see using the words up here. So as you can see, we have represented here a body cell of the female parent. So hopefully you remember that body cells are also known as somatic cells. there would also be the body cells of the male. There's going to be a small portion of those body cells that are going to undergo a process to produce the gametes. And notice the chromosomes in the middle, in the nucleus, that there were 4 originally, and now there are only 2. So we've gone from a diploid somatic cell to a haploid gamete, okay? That's the sperm and the egg, and then when those 2 combine, they are going to become diploid again. That's the process of fertilization. So we go from diploid to haploid back to diploid. All right, let's look at this next diagram. And after we look at it together, pause the video and you can answer these questions below. First one saying, is the cell shown haploid or diploid? And make sure you can explain. Well, remember that diploid means there's going to be a full set of homologous pairs. And so if we look carefully at this diagram, there is a full pair right there. Here's another pair. And then here's a third pair. So we do have homologous pairs. If we have homologous pairs, then it's going to be a diploid cell. Then is it somatic or a gamete? Well, if it's somatic, it's going to be 2N. It's going to be diploid. If it's a gamete, it would just be 1N. There would only be one of each of these homologous pairs. So somatic cells have the full set. Gametes only have half. So in a human cell, if the diploid number, which is represented and modeled by this symbol 2N, if the diploid number is 46, then you would cut this in half and you would get 23 for a haploid cell in a human. So how do we know how many chromosomes are in a particular cell? Scientists will prepare what is called a karyotype, which is an organized visual of the organism's chromosomes. These are prepared from cells that are in the middle of the cell division phase, the M phase, when the chromosomes are all condensed and coiled and you can see them. So this is what it would look like in a real cell. And that's why chromosomes are often represented with these X shapes. So some chromosomes are known as autosomes. Autosomes are going to be all of the chromosomes in the organism that are not related to determining gender, okay? So they're everything except for the sex chromosomes. So then the sex chromosomes would be that one pair that's going to help determine gender. It's going to contain genes that determine gender. So in a human cell, there's going to be 22 pairs of autosomes, and then there's going to be one pair of the sex chromosomes. In this particular karyotype, it represents a human cell, and this one is showing a female. Okay, so you're going to know it's a female because you have 2 X chromosomes. And when there are 2 X chromosomes, they are true homologs. They're the same size, same banding patterns under the microscope, etc. If it was a male, you would not have a true homologous pair. You would have what's known as an XY. And it's actually really a dramatic size difference. The X chromosome are really big and long and lots of DNA on it, and then the Y chromosome is this tiny little chromosome. So it's not a true homologous pair, but that would be what you would see in the karyotype for a male. Okay, next question. Are the chromosomes from a diploid or a haploid cell? Well, look at the karyotype right there. You've got a pair. If you have the complete pair, it's going to be from a diploid cell. And then finally, are the chromosomes made of single or sister chromatids? Well, here we go. We've got 2 copies per chromosome. So this must be sister chromatids. And again, like I said before, this is coming from the M phase of the cell cycle. So it's after G1, S, and G2. That's why we have sister chromatids. The DNA has already been replicated, and the DNA is nicely coiled and condensed and really easy to see under the microscope. And that's what happens during cell division. Everything gets packed up for the move. Here's the procedure of karyotyping, just in case you're curious. And so it would involve taking a blood sample and then culturing it in the lab for a few days, adding some chemicals to fix the cells onto a slide, and then adding some stain to them so that you can actually see the DNA. And again, this would be the metaphase spread. So during cell division, that's when the chromosomes are all coiled like this and easy to see. And then, now there's computer analysis to look at this, but you would identify each pair of chromosomes, and that's a way of determining whether it's coming from a male or female. And it's also a way of determining that the chromosome number is normal, because there are some situations where there can be an abnormal or atypical number of chromosomes, which can lead to certain disorders. Okay, so finally I'd like us to review the relationship between a homologous chromosome and an allele. Where do alleles come from? How do they occur? So remember that when we talk about a homologous chromosome, you've got a pair of chromosomes that are not connected. One comes from the male parent and one is going to come from the female parent, and you would find this complete pair in a diploid cell. And those homologous pairs could be made of sister chromatids, so there's actually 4 sets of DNA in this picture, or they can be made of single chromatids, and that would be before DNA replication. In any case, they're still homologous pairs because they're the same size, they have similar banding patterns, and they're going to contain similar but not identical sequences of DNA. So those differences that make them not identical, that's what's known as alleles, because that's where you get alternate versions of genes. So maybe right here you have a sequence that codes for a particular protein in a flower that gives a purple pigment, and then a slightly different version of that DNA sequence would not code for a functional protein that would help make the purple pigment, and therefore that contributes to that flower being white. And then we're going to look at the interaction. If 2 homologous chromosomes have different versions of alleles, we'll look at that and see which one will the flower actually be. Will it be purple or white? But this is where those differences come from. It's going to be where you have a slightly different DNA sequence on that gene right there. So where do those alleles actually come from? How do they occur? Well, they occur through mutation. Whenever that DNA sequence gets changed, either just through random error or DNA damage from some toxin or chemical exposure or UV light, or just, again, it could be a random error during DNA replication, you'll get a slightly different sequence of DNA. And when you get that new sequence of DNA, now it is an allele because it is an alternate version of the gene. Okay, we're going to pause right there, and I will continue in the next video. We'll be looking at the actual process of meiosis.